Ladies, gentlemen, my name is Marc Léon. I'm a physician in Marseille, France, and that's my pleasure to thank you for attending to this webinar organized by the CIS section of EasyChem, and that's our pleasure today to welcome Professor Richard Hotchkiss from Washington University in St. Louis, and Richard is going to tell us his research history about sepsis. So I think that I co-chair with Elisa Damiani from Ancona, and I guess that Elisa can have a question for Richard.
Yes, sure. Hello, everyone. I am Elisa Damiani. I am an associate professor of anesthesiology at Università Politecnica delle Marche in Ancona, Italy. And so let's start with my first question to Professor Hotchkiss. It's an honor to have you here today. So Professor Hotchkiss, with your studies, you gave a crucial contribution to the understanding of sepsis pathophysiology. Can I ask you, when did your interest in sepsis start, and how did you become aware that it was not just hyperinflammation and cytokine storm?
Yeah, thank you very much. First, it's a real honor to be invited to give this talk, and have enjoyed my collaborations with a number of investigators, both in France and Italy and Germany, and I've learned a great deal from them. So my interest in sepsis, I did internal medicine training and always found the ICU to be the place where the rubber meets the road, so to speak. You know, you really have to be on your game. There's life and death situations. The ICU attracts the brightest patients, I think, because you have to be ready. I call it the crucible. It's a place of intense testing. You have to be able to act quickly, decisively, and you have to be willing to work hard. And all the people in the ICU, you know, my wife's a former ICU nurse, and just it really attracts outstanding people. And so that's how, you know, in ICU I got interested in it. And sepsis, well, it's the #1 cause of death in the surgical ICU, many ICUs, and was really interested in some of the metabolic changes, which I'll talk about. And then, you know, you go down a lot of wrong paths, and then you learn, you know, what really is going on, and that's kind of the some of the basis of my talk today.
So thank you, Richard. So that's really a pleasure well, really happy to have you with us today. That was an old dream for me, because we worked together 20 years ago now,
Yeah.
St. Louis, and you learned me a lot during this year, running in the ICU, and even in the lab where we were close each other. So the floor is yours now for your presentation, and
Yeah.
we have 25 minutes of happiness with you.
Yeah. Thanks a lot, Mark. And I remember you were an outstanding clinician, and it was a pleasure having you, and have enjoyed following your work and that for these number of years. So I'll get started here, and we'll make this like an overview. So when I originally got interested in sepsis in the 1970s, the theory was that sepsis was an energy deficit problem. And this made sense. There was increased lactate, metabolic acidosis. The theory was that part of it, there was not enough oxygen being delivered to the tissues. Also, there are a number of studies showing that there was mitochondrial dysfunction. So this was what was felt to be sepsis, an energy deficit disorder, either from cellular hypoxia or mitochondrial dysfunction. So I got very interested in NMR spectroscopy because, you know, the ATP, NMR, 31P NMR spectroscopy tells you about the levels of the ATP gamma alpha and beta ATP, it allows you to tell about the intracellular pH of the cell. And I figured this was a really good way to determine about, is sepsis an energy deficit problem? So I was recruited to Washington University and was fortunate enough to work under Dr. Joe Ackerman, who was chair, and he did landmark studies published in Nature using NMR spectroscopy to determine in vivo, did some of the first studies in animals looking at the NMR, the energy levels of the cell. So we did a series of studies with NMR spectroscopy in rat brain, looking at 31P as the brain losing its energy. We looked at heart and we looked at muscle. And to my surprise, there really was not any loss of cellular energy, that is the ATP levels, until the animal was basically agonal. We wrote about this reevaluation of the role of cellular hypoxia in this, and you can see there really was not energy deficit, and a lot of the increase in lactate was due not to cellular hypoxia, but to an increase in glucose uptake in this. There was subsequently a study done at the NIH by Bob Balaban. He was an expert in this, where they looked at myocardial energy metabolism in a canine model of sepsis. And basically they found this exact same thing in a larger model in dogs. And they said that the canine model of sepsis, even though the heart was depressed, poor function, it was not due to low ATP or mitochondrial dysfunction. So I struck out there. So what do we go next? So, as you know, also at that time, there was the concept that Lewis Thomas in the New England Journal popularized this, that the microorganisms that seem to have it in for us turn out to be rather like bystanders. It's our response to their presence that makes the disease. We're more in danger from our body's response. In science, it was called an immune system gone haywire. Working in the ICU, we all do see patients like this. With group A, patients will just come in with fulminant sepsis and they'll die, 24, 48 hours, despite your best efforts. Meningococcemia, C. diff toxins. And so the theory was blocking the host response would improve survival. So we then undertook some series of studies using endotoxin or live bacteria. This made sense because in the animal models, when you inject endotoxin or live bacteria, it's like an atomic bomb, occurring on the animal. And the levels of at the same time, TNF alpha was discovered in the 70s and 80s, and other pro-inflammatory markers. And if you block them, the animals survive. So this made sense. So the two hallmarks of sepsis were the hyperinflammation and this protein breakdown. If you work in the ICU and you treat these patients, you can see their muscle mass, just they sort of lose their muscle mass so dramatically over that period of the hyperinflammation. So I started thinking, there was some data in the literature that a lot of these proteases and things that break down the muscle are calcium regulated proteases. So I started thinking about, well, maybe if we block the calcium mobilization, we can improve mortality and prevent this muscle breakdown. So this is interesting. Let's see if I can move this here. There we go. So I was fortunate to work in the laboratory. Again, a great collaboration with Dr. Irene Carl. She ran the metabolism lab and taught me a great deal. So I was anesthesia trained as well as internal medicine, and I knew about Dantrolene. Dantrolene, as you're aware, is life-saving in malignant hyperthermia. That's a disorder of calcium reuptake in there. And Dantrolene is life-saving in the patients that have malignant hyperthermia. If you don't give them Dantrolene, there's about a 90% mortality. But giving the Dantrolene, really, it's a remarkable life-saving advance. So we did some studies where we had a endotoxin model in mice, and we gave the endotoxin, and then we gave Dantrolene. And Dantrolene decreased this muscle protein catabolism by over 50%. Here you can see the tyrosine release in the control muscle. In the septic muscle, it was almost doubled up to, you see the yellow highlighted, 2.4. And then in the treated with Dantrolene, it brought it back down to normal. So it blocked muscle protein breakdown. And on the right-hand panel, you see there is a marked improvement in survival in the mice that got endotoxin that were treated with Dantrolene. Notice the large number of mice in the study. So we also showed in a subsequent paper in Shock that Dantrolene also decreased the levels of TNF alpha and interleukins in this model by about 50 to 60%. So, I mean, we were really excited about this. You know, we talked to Procter & Gamble, who made the Dantrolene. They patented, and, you know, we were quite excited to move. This was a therapy. You know, we're all congratulating ourselves. And there was even another study by Wu and Yin, and they substantiated our work in an endotoxin model, again, an endotoxin model, and improved survival. So I also knew that Mitch Fink, who was a chair of at University of Pittsburgh Medical Center, was testing Dantrolene in a different model. They tested it in the cecal ligation and puncture model. This is a model that's much more reflects what happens in patients. The mice get polymicrobial sepsis and die over this. So what did Mitch find? I remember I came up to Mitch at the meeting in Shock, said, Mitch, I know what's going on? You know, did the Dantrolene improve survival? And he said they died like flies. Okay, so the exact opposite. It decreased survival in this other model, bacterial sepsis. So what was my response to this thing? This was my response to it. I said, oh my gosh. I mean, I remember being at the meeting. I went back to my room. I laid down on my bed, looked up at the ceiling, you know, because I was so, you know, disappointed with it. But it was really this thing, this kind of counter in the thing that helped me shape my thinking, that turned me around to help figure it out. The answer to this question, why did Dantrolene improve survival in the endotoxin model, but worsen survival in the CLP model? And this was critical. So in the endotoxin model, it's a cytokine storm mediated. And so the levels of these pro-inflammatory cytokines are very high in the nanogram level. But in the CLP model with this, the levels are only in the picogram, and they're much lower. And you don't have any living bacterial organisms in the endotoxin model. You don't have to, you know, kill the bacteria. Whereas in the polymicrobe, you have you have multi-polymicrobial sepsis. So, as I said, the immune effector cells in the CLP model need calcium to function. You know, if you block calcium, which Dantrolene was very good at, the lymphocytes don't make cytokines, the neutrophils and monocytes don't move, phagocytosis is impaired. So blocking that impairs immune function, but it's life-saving in the LPS model because you decrease the cytokinemia. What is the case in patients? So I like this slide. Just because you're traveling down a well-trodden path doesn't mean you're heading in the right direction. This was another thing I learned here. In Sherlock Holmes, a brilliant detective, it's a capital mistake to theorize before one has data. Insensibly, one begins to twist facts to suit theories instead of theories to suit facts. And I learned this the hard way. So back to the drawing board, what we do. So in thinking about this time, there was a new cell death description. Apoptosis started coming into the literature. And I was very interested in this because, you know, in the ICU, when you look at your patients, they have this organ dysfunction, but there's not a lot of the enzymes released that you typically see with cell death. Like a lot of these patients, their kidney function had stopped working, but there wasn't a lot of CPK released or other things, and the liver had stopped working, but there wasn't a lot of ALT or AST being released. So it kind of made sense, all this organ function without this. So we had a review article in the New England Journal where we looked, on cell death and pathways of cell death. And on the left-hand side here, you can see the apoptosis, the cells become shrunken, the cell membrane integrity is intact, and you get this nuclear fragmentation and compaction, whereas with necrosis, the cells swell and they lose their ion channels, they open up, and so the cells swell and there's leakage of all these proteases and lysozymes, which causes a lot of inflammation. I was again very blessed to have Paul Swanson, who was head of anatomic pathology. He ran the histochemistry lab, and his specialty was in cell death. And we collaborated for many years on this. And we went back to the mouse CLP model, and we harvested the tissues from different organs and gave them to Paul to do EMs on and other things. And here you can see in the upper panel is the normal looking thymocytes. The chromatin is very well spread out. The cells are nice and big, but in the septic thymus, you can see the nuclei become very small and the nuclear chromatin is very compacted. This is not a subtle finding. You can see on the H&E staining for the thymus from the septic mice versus the sham mice, there's just incredible widespread death, of large fields, apoptotic death in there. And here's for thymocyte apoptosis in the mouse model, the CLP model. It's very interesting. You see the cells sort of die in these islands here. And this was a thymus from a neonate who died on the right-hand panel of necrotizing enterocolitis. There's very similar here. This is an electron microscope from a patient who died of Pseudomonas pneumonia. And again, you see these stellate, these crescent-shaped moon things. And here's in the mouse model, this is a tunnel. Another way it labels these nuclear apoptotic cells, and you can see the intense. So we went and we did autopsy studies in the ICU. It was an amazing study. We talked to the families. Being an ICU physician, you get very close to a lot of the patients, or you should. You should talk to them all the time in this. And it was amazing how many times when talking to them about, after their family member died, about allowing us to get tissues to figure out this. And so many of them, it provided some sense of meaning for that their loved one's death could help somebody else. So we did this autopsy study in the ICU. We had good surgical colleagues, and generally anywhere from 30 minutes to 90 minutes after they died, we harvested, did a mini autopsy, did a laparotomy, got spleen, liver, kidney, then cut the diaphragm and GI tract and got heart and lung. And here you can see on the left-hand panel, it's the dark staining foci are B cell follicles. Okay, these are just 6 slides that are stained for B cells. The ones on the left are trauma. These were patients that had their spleens removed for trauma. They were young, healthy patients in general. So this is what you would look like. But in the septic patients, you can see they've lost so many of their B cells. I said, the best way to show you this is just put the slides up. This is just the slides, okay? Now, CD4 T cells on the right-hand panel, you actually have to look at with a microscope. All the brown staining cells on the trauma patient are CD4 T cells, but you can see the loss of CD4 helpers, CD4 T cells on this. And Joe Carcillo at the University of Pittsburgh repeated our study really almost identically in pediatric patients that died of sepsis in a paper. And he showed the exact same thing that we did. The B cell depletion in pediatric patients dying of sepsis and CD4 T cell depletion. Interestingly, the other organ that died and with sepsis was the lining of your GI tract. The arrows show these compacted nuclei, and your GI tract normally gets sloughed every 3 to 5 days. So what happens is it just accelerates the process. And these are classic, the era's apoptotic ones. Here you can see on the right-hand panel, just that villus just blown away with apoptotic lymphocytes. So we published this paper on this with 20 patients and showed heart, lung, liver, and spleen. Apoptosis was the predominant mechanism of cell death in lymphocytes and intestinal epithelial cells. And there's marked depletion of CD4. And we said at that time, we thought that sepsis-induced depletion of immune effector cells would result in immune suppression was likely a key pathogenic mechanism. Interesting, the paper was rejected by the New England Journal, JAMA, and JCI, and we published it in Critical Care Medicine, where it's been cited over 1,800 times. So don't give up. Okay, so the key question is the loss in lymphocytes an epiphenomenon, or does it play an essential pathogenic role? We've known for a long time, a lot of studies, correlation of absolute lymphocyte counts correlate with survival. Here on the green line, if the lymphocyte counts rise, you survive. If they stay low, higher incidence of mortality. So just because you have a low lymphocyte count and it correlates does not mean it's causative. Correlation does not prove causation. So how do you prove it? Is the loss in lymphocytes an epiphenomena or a key pathophysiologic mechanism? So in the 2002 Nobel Prize in Physiology was awarded for the studies on cell death and apoptosis. Bob Horvitz was one of the leaders at MIT on this. And he was working because, again, I mean, I was so fortunate and blessed with having great collaborators. Stan Korsmeyer was at WashU at the time, and he really was instrumental in defining this relationship between anti-apoptotic BCL2 members and family. And there was the balance between the pro-apoptotic BCL2 and the anti-apoptotic that really determined the life or death of the lymphocyte. In fact, Bob Horvitz, who won the Nobel Prize, said he always expected Stan to be on the award with him. Stan unfortunately died of cancer after this, but we collaborated, and he had mice that overexpressed BCL2 in these lymphocytes. And so I said, well, I'm going to try. I didn't expect anything. So in these mice that over here, you can see the cell death in the lymphocytes from the septic control mice. And here was the histology from the BCL2 overexpressing mice. They were almost totally protected. And here you can see on the DNA agarose gels, this is classic, this DNA laddering for apoptosis. And there was, I couldn't believe it. When the first time our Gabby, our laboratory technician showed me this, I said, well, something's wrong. She repeated it. And I said, this cannot be this good. So she did it a third time. But I said, it's real. So we subsequently showed, and two other groups did too, that mice that overexpress BCL2 have an improved survival in sepsis. So this was quite encouraging. If you can prevent the death of the lymphocytes by BCL2 overexpression. So the question is, if BCL2 prevents sepsis-induced, how do you increase BCL2 in patients? So fortunately, there is a cytokine, IL-7, IL-15 also does this. It increases BCL2 through STAT5, and the BCL2 blocks this mitochondrial-induced cell death. We tried IL-7 in mice, and we showed that by giving IL-7, you can increase the BCL2. This is by flow cytometry staining for BCL2 and lymphocytes. You can see there's a very good increase here. And also shown in the CD4 T cells, mice that get treated with IL-7 have a big increase in the expression detected by flow cytometry. And furthermore, it prevents the apoptosis. Here's the loss in splenic, in total cell counts and CD8 T cell counts, which is prevented or diminished this. Again, we did studies in septic mouse model and showed that IL-7 administration improved survival. There were 2 independent labs that showed that in different strains of mice. So it was a consistent finding IL-7 works. So conclusions and lessons learned here. Mouse models of sepsis can be highly misleading. I was misled by the endotoxin model. Mouse models can be highly informative. I was definitely taught a lot, learned by looking at the apoptosis in the mouse model, and it reflected very closely the patients. The other thing I learned was follow the problem and don't get wedded to the technology. I've seen so many people get focused on the technology. Okay, get your problem and follow wherever it goes. The most important thing I can probably tell you is combine translational studies if possible. It was going to the bedside and doing those human autopsy studies, which really opened up the field for this. Continue to question and challenge your underlying hypotheses, because our first 2 hypotheses were dead wrong, and it took a while to get. Be persistent. I mean, if I hadn't been there were times I thought about saying, you know, it's too frustrating, you know, in trying to get grants and things, but, you know, finally seek out great mentors. I was so blessed with wonderful mentors. Hire great people. Treat them well, they'll like to be treated, and make them feel part of the mission. I have to give credit to my laboratory people. A lot of people at Washington University say I've got the best people in the lab. Actually, it's mostly the people in my lab that say I have the best people at WashU. And I think I'll stop there and happy to answer questions.
Thanks a lot, Professor Hotchkiss, for this beautiful presentation. So the results of your research introduced a new paradigm in sepsis treatment, from just targeting the pathogen to targeting the host immunity together with the pathogen. However, this seems like a very difficult task, given the high complexity of the cellular mechanisms of immunity and their changes over time in human sepsis. So how to move on? How to personalize immunoadjuvant therapies in septic patients? How to design future clinical trials?
Well, that is a great question in the heart of the matter. So I think there's a number of ways to approach it. I do believe that doing functional testing of the immune cells is very important. In other words, there are a lot of people looking at transcription, gene transcription to try to determine if they're immune suppressed in the hyperinflammatory or immune suppressive phase. And that may come. I believe that functional testing of the immune system is the way that's be most helpful with this. In other words, some of the studies that are being done and that we're doing with a large NIH-funded grant is where we take blood from the septic patients and we will stimulate that blood with CD3, CD20, something that stimulates T cells. And then you quantify interferon gamma production over a certain period of time to tell you about the adaptive immune system. We also stimulate it with endotoxin and look at TNF alpha production. Okay, there are a number of ways to do this. I know that bioMérieux is looking at this. The other company, Roche, is looking at this. I think that these functional tests to see are the best way to tell whether you need this. And we've published a number of papers on this. This has been a major focus of our lab. We have papers in JCI Insight and Journal Immunology showing that patients, when you do these tests for their production, functional effects, they predict outcomes. Furthermore, we've published a number of case reports. We've shown that patients, for example, that have these intractable fungal infections are highly immune suppressed. And when you give them drugs like IL-7 or checkpoint inhibitors or things, their immune function corresponds and correlates with their ability to clear the infection. So I think that function is the way to go. There's some nice editorials in some of the critical care literature about this. I think that's one way. Now, the other thing I'd like to say is there are some clues that are pretty obvious to me. As ICU clinicians, you need to use all the input. So about if someone comes in to the ICU and they're in septic shock and they're hypothermic, okay, this patient is not mounting a good response. Their mortality is higher. It's typically patients that are immune suppressed, like renal dialysis patients or elderly patients or things like that. Okay. I think it is unwise to give that patient corticosteroids. Okay, there are data. Hector Wong had some data on this, that pediatric patients in septic shock, he had a gene expression panel. If they did the gene expression panel and the results looked like immune suppression, and you gave them steroids, they had worse outcomes. If you, on the other hand, if they had this gene expression panel and looked more hyperinflammatory and you gave them steroids, they did better. So I think that the functional studies, but also used your the clinical factors in here. I think there are a lot of patients walking around who have baseline immune suppression. Patients that are elderly have immune suppression. Patients with COPD, the renal dialysis patients are immune suppressive. I think that's in the ICU for secondary infections. I think if the patients are getting infections with a lot of these less virulent pathogens like Acinetobacter, Stenotrophomonas, fungal infections, I think those patients that get these hospital-acquired infections are frequently, that's evidence of immune suppression, because normal immune systems don't get those type of infections. The other thing I think I would say is, and I'd like this to be very, and if you look at the realism trial, which is a great trial, done by the French, multiple centers there, supported by bioMérieux, they showed that in the first days, somebody that comes in with severe sepsis, their cytokine levels, TNF-alpha, IL-6s, are higher than patients that come in with trauma or have major surgery. Okay, however, by 72 hours, in other words, after that, the levels, these high cytokines, will come down. Okay, so that they're very comparable to levels of trauma patients and patients that had major surgery. So I think if you really needed to do immune therapy, to boost the immune system, after that 72 hours, depending upon that tends to also correlate with it. You're usually way down on their inotropic agents and not having as much trouble with keeping their blood pressure up. So I think temporarily this, but I think the field still needs to evolve to develop these tests, and a number of groups are working. I'm sorry for the long-winded response. It's the most important part of the question there.
Richard, there is a lot of studies about phenotype and how phenotype can change the outcome of patients. What do you think about these studies, and what is for you the best biomarker for the future?
Yeah. So the thing is, with some of the companies we tried to get them to do it, I wish they would. They have these, the instruments are currently that they measure CBC or chemistries or things like. They're in the hospital already. Okay, some of these companies could do this, where they just add, the stimulants to the patient's blood, and look at these cytokine productions, either interferon gamma or IL-6. And we're actually in discussions. I wish you guys would, the more people that beat on them and tell me you got to do this, the better. Okay, I know there are other assays such as a monocyte HLA DR, expression percent that Hume's working on. I think it needs to be something very simple. The great thing about, I think that if you look at the oncology literature, the things that are working best in the oncology literature are things that work to stimulate T cells. Okay, because the T cells, the CD4 helper cells, they are sort of like the orchestra conductor. They release the cytokines, which will engage the innate immune system as well. Oncology field, I've had trouble. They're getting there. The things that boost, the innate immune system, monocyte, macrophages, and they'll get there. Some encouraging ones, but I think things that looking at your T cell function, is critical. Now, I won't get into absolute lymphocyte counts. I think they could be important, but that's another whole thing to talk about. It's not always cell death why they're low. But the test needs to be, people are wanting a test in, an hour or two to decide. I don't, a lot of these patients are septic for days. So I don't think it's as critical to have something, that, tells you like that. It might be for giving steroids, no go, but I think that looking at adaptive immunity, T cell function, and interferon gamma, if I had to pick one, I would say that. What will probably happen is they will get the gene expression patterns that will say this gene expression pattern correlates with patients that make a lot of interferon gamma, and this gene expression pattern correlates with patients that are immune suppressed. And so then maybe you can just do the gene expression thing. But I think looking at functional studies with T cell interferon gamma production would, if you really force me to say that.
I have another question for you. Should we include immune dysfunction in the SOFA score? And how would you do so?
Yeah. So I think here absolute lymphocyte counts are helpful here. Patients that are septic, that whose absolute lymphocyte counts are, in the normal range, generally have a very good, they don't have the high mortality. Okay, that's a good sign. If they're so the CBC with differential and following that is helpful. On the other hand, if it's a very low absolute lymphocyte count, there's two reasons for that. Okay, one is patients with trauma actually on the first day or two have just as low absolute lymphocyte counts as septic patients. Why is that? That's because with trauma, the lymphocytes get activated and they express these markers, these adhesion markers, and they leave the circulation, okay, in the first 24 hours. Now, the trauma patients, if they don't get septic, by 72 hours, their lymphocyte counts are back up to normal or low normal. But the septic patients, they're stay low. So I think the clinicians, I really am a believer in following the absolute lymphocyte counts for the progression of sepsis, or if, development of secondary infections. Okay, I don't know about putting it in the SOFA score, but something clinicians should really follow the trajectory of it. It's a simple thing to follow for this. It's part of the piece of the puzzle on this.
Okay, thanks. Mark, do you have any other question for Professor Hotchkiss?
Yeah, just a simple question. Do you still believe in interleukin 7, or you believe rather in checkpoint inhibitor, or what is for you the best drug today with maximum chance of success in sepsis? It is one drug for all patients, or we have to personalize?
I tell you, Mark, I really believe in interleukin 7, okay, because it's safe. We have a paper that's returned with revision. Bruno Francois was a big leader. So was Manu. On this, it's 109 patients, randomized, double-blind, placebo-controlled trial in COVID patients, critically ill COVID patients, and there were nice improvements with a decrease in secondary hospital-acquired infections. And strong trends towards. I like that drug because it's safe. It was well tolerated. Okay, there was one patient that had to be re-intubated for overnight and extubated, left the ICU, did well, but they didn't see real big bumps in the cytokines. I think it's a great drug, and we have a lot of case reports with it. It's being used in oncology. There was just an oncology trial, and it's effective in that. I like that. I think the checkpoint inhibitors, I think they have efficacy. That's not as effective with multiple errors as IL-7 is, and so I would go for safety. I would go with IL-7, and we're moving forward with hope to begin large clinical trials. We've done some studies in sepsis and encouraged about that. But I think looking in the future, there's going to be a number of drugs. I think IL-15, depending upon the type of pathogen, if you have an intracellular pathogen, IL-15 may be better because it stimulates the NK cells and monocytes to kill the extracellular ones. IL-7 maybe. It's an exciting area, and I hope I live long enough to see the field evolve in that direction. Thank you very much.
Thank you so much, Professor Hotchkiss, for this wonderful talk. We could keep asking you questions for hours, I think, but I'm afraid we must come to an end now. Personally, as a junior investigator facing all the difficulties of doing research, I can say that people like you, with your passion, your perseverance, and also with your humbleness, are the ones who can really motivate us to go forward with our studies. So it has been an honor for me to be part of this webinar and to talk to you today. I wish to express my sincere gratitude for your contribution to this activity of the ESICM. And thanks, Mark, for co-moderating this with me today.